Welcome to Ask the Expert. It's a brief, informative, and lively discussion with experts in type 1 diabetes and interdisciplinary research. We're recording this event and we're going to post it on the Sugar Science YouTube channel shortly after the presentation. If you have questions for our guests, please feel free to enter them in the chat or raise your hand at the end of the presentation. And today we have as our guest, Patricia Aparicio-Domingo. Correct me if that's not the right pronunciation. She's PhD. And she is Abcam's research lead for immunology. She's really responsible for managing the development of products in the immunology world and infectious disease world. And prior to joining Abcam, Patricia was a postdoctoral fellow at University of Lausanne. She studied the role of stromal cells in regulating adaptive immune responses, and she holds a PhD in immunology from Erasmus University Medical Center, also an MSc in biomolecular sciences, and she's co-authored numerous scientific papers, including publications in the Journal of Experimental Medicine, a European Journal of Immunology, Frontiers in Immunology, and the Journal of Innate Immunology. So the title of her presentation is really supporting type 1 diabetes research with sustainable, validated, and consistent reagents. And we all know that antibodies are critical for, really validating your work. If you don't have the right antibody and do the experiment, it's a lot of wasted time and effort if no one else can validate that because of the antibodies, weakness, I guess you can call it. And I'll talk just a little tiny bit about Abcam. Abcam is a global life science company, and it really develops, distributes, and identifies high quality biological reagents. And I think they also, they're innovative. Abcam is also innovative in that they partner with life sciences organizations. They intend to co-create novel binders for use in drug discovery, in vitro diagnostics, and therapeutics. And it's very interesting that they've just sort of partnered with Icaros. And so I hope that she'll touch on that a little bit here. So welcome, Patricia, and thank you for joining us.
Thank you so much, Monica, for the kind introduction, and of course for the invitation of being part of this Sugar Science initiative, which I think is really interesting. So I'm gonna share my slides, if that's fine.
Sure.
Let me see. Let me know when you can see my screen, please. Can you all see my slides?
Yes, that's perfect.
All right. All right. So, yeah, so thank you for the introduction. Indeed, I am Patricia Aparicio. You got it almost right. So I'm the research area lead for immunology, and today is really my pleasure to speak to you about how we at Abcam are having an impact in type 1 diabetes research by providing validated and sustainable, consistent antibodies and reagents. So I joined Abcam last October, and in addition to having an impact in science, something that really keeps me excited at Abcam is our commitment to increase scientific reproducibility. As you just very well said, we're having this crisis of reproducibility, where scientists in the different labs are not able to replicate the data, and this is mostly due to bad reagents. So let me kick off by just talking a little bit about Abcam. So Abcam was founded in 1998 here in Cambridge. It was a small antibody company that really aimed to provide high quality reagents, antibodies for scientists. And since then, we've been growing a lot. So in the past 20 years, we have now 13 different locations across the globe with more than 1,600 employees and with more than 90,000 products. So just to give you an overview, we are serving the different sectors. Of course, we are mainly focus until now has been academia. So we've been providing reagents and antibodies for universities, research institutes, but we are also having a sector, and we are developing products for the pharma industry as well as for the diagnostic development. So um, so I said we have 90,000 products. So this includes, of course, antibodies, cell lines, proteins, peptides. However, we are mostly an antibody company. So this is really our expertise, and this is our experience. Basically, we have a really well developed procedure and methodology where we have the different teams working on a specific subset and a specific methodologies. So we have a very specialized group of scientists working on the protein team, working on the design of the antigens, the immunogen, and selecting and doing research on the protein and the target selected. We have a cell science team working on the production and the expression, purification. And of course, we have scientists working on the development and the cloning and the screening of the antibodies, just to select one final clone that will pass to characterization and to final quality control. So this is just an overview of the whole process. Of course, it is very, as you can imagine, there's many different aspects involved. But something that is important for us in Abcam to achieve high quality monoclonal antibodies is to follow some sort of guidelines. And this is what we call the pillars in Abcam for antibody validation. So everything starts by doing the study of the research on the target, so the target background. This means understanding really what kind of protein it is, how it is folded, how can we design it, what are the conformational epitopes, is it linear, is it a three-dimensional epitope, really understanding how this protein is going to be. We have in place the right methodology, so we make sure we are following the protocols that are highly optimized, and they are run by scientists that are trained, and they are running the experiments in state-of-the-art facilities. And we are doing these assays, these tests, using the right cell types. And this is really important for some specific antibodies. So we are using primary cells when it's required and when we can. And sometimes we have to use diseased tissue for immunohistochemistry, for instance, and we compare it with healthy tissue. And when possible, and we are also using sometimes biopsies from patient samples.
So Patricia, how often do you have access to type 1 diabetes patient samples? I mean, is that something you really, interface with in terms of this workflow?
Yeah, so we do have some collaborations and some agreements in place with different biobanks, for instance. And as I will mention in a bit, we are working closely with collaborators from different institutes. Which that means is they can help us testing on the right settings. So sometimes we cannot really get these tissues from patient to our lab, but we can give the antibodies to the scientists working with these tissues. Sometimes it's human tissues with diabetes patients, really, but sometimes it's with a murine model with a knockout system.
It seems like, T1D Exchange is right across the river there in Boston, right? And so they would be a really great help to source some of these patient samples. Am I right?
Right. It's all about finding the right places, the institutes. As I said, biobanks. We're working with some of the hospitals just next to where Abcam headquarters is. So it is really this network of scientists providing different tissues and allowing us to do our job better, really.
Great. Viva collaboration.
Yeah, it's really an active collaboration, and we really depend on collaborators and scientists as we don't have all the expertise, of course, and we really need all the different scientists. Yeah.
Yeah, that's
So
powerful.
yeah, absolutely. Yeah. So this is about primary cells or disease tissues, sample biopsies or patient samples. But we also make sure when we develop our antibodies that we are running the assays under the good conditions on the right settings. So what this means is that sometimes we need to stimulate, for instance, we have to use some cytokines, or we need to use some chemicals to either induce the expression of the protein or to downregulate the expression. And we have to choose the right application. Sometimes it's designed for Western blot or for flow cytometry. So we really make sure we are having the whole picture. And something also extremely important when designing and developing antibodies and validating these antibodies is the use of the right controls, and especially, so what we use is a knockout validation system. So we're knocking out or knocking down in some cases the protein of interest to really look at the specificity of the antibody. And we also check other cross reactions, for instance, with isoforms or with family members, to make sure that the antibody is really specific for this target.
Great.
Furthermore, yeah, because we're using recombinant antibodies, of course, we avoid all these issues with consistency. We have really robust and consistent antibodies. And as you very well mentioned and highlighted, the role of the collaborators and external partners are crucial. This is critical for the development and tests and validation of the antibodies. So as I said, we have external collaborators from scientists in academia, but also in pharma, in big pharmaceutical companies. They're also helping us with the testing. And we have collaborators that are giving us data before publication of the antibody, so to make sure that the antibody we are launching is of the high quality standard that we expect to, but also after publication. And we achieve that by using the Abcam system, which is in our website. So it's just a way to get feedback from scientists, either positive or negative. We value both feedbacks to improve, of course, and also positive to make it more consistent and more robust.
That's great. So how do scientists, type 1 diabetes scientists, access the AbReview portal or, system?
Yeah, this is it is straightforward. You basically need, so once you have a product and you have tested one of our products, let's say one of these recombinant antibodies, monoclonal antibodies, you just need to go to the product page, and then you can submit an AbReview, and then we will ask a couple of questions, like about what kind of controls do you use, and what is the concentration of the antibody, these sort of questions, like very basic to understand that the experiment was done properly. And that's it. And then we just display that information on our website, and it's public, and everybody can see it.
So it's kind of like a Yelp for antibodies. And how much traffic do you have from the type 1 diabetes research community? I mean, are people coming in there, or is this a chance to invite more people to do so?
Exactly. Well, we have quite a lot of AbReviews in metabolic diseases and in diabetes. Our portfolio for diabetes is quite extensive, I would say. So we have some of them. I will just give you some examples now, but some of them are getting really good reviews, meaning that scientists are really happy testing it in all the different applications, because some of these antibodies are suitable for different applications, so we can get all these comments and feedback. So
well, we did our own little due diligence here with reaching out to the community, and the Abcam name came up, most often as sort of the trusted go-to, just sort of that feedback was pretty impressive.
That's really good to hear. we believe that our antibodies are really make the customers and the scientists really happy. We really make the effort to validate them carefully. We characterize them, and what we see so far is that we are getting really good feedback.
Yeah.
And of course, that generates more interest for the scientists, and they are, we have like a rating system with stars. So if you like it and it worked perfectly in your application, in your experiment, you can give us like a 5-star review, and that will, be a way to promote somehow this product. So it's a really great system to get the opinion of the scientists. So I encourage you to to have a look if you have the time. So basically we have these different pillars to make sure we have highly characterized recombinant monoclonal antibodies. And the important thing is that this translates into reproducible data, right? So this is finally the main goal and the objective. And I think Abcam in that sense is doing a great job supporting reproducible data. So as I mentioned, because our antibodies are so specific and designed carefully, we are making sure that we are only recognizing a single epitope, and they're highly reproducible as a consequence, with no variation between the different batches. And in here we have just an example of an antibody, recombinant anti-PD-L1 antibody. So these are 6 different stainings generated with different batches of this antibody. And as you can see, the staining pattern is pretty much the same, is almost identical. So there is no variability there, and which is what we expect, of course. And we also have this high and improved sensitivity and specificity. And then because we're using recombinant, so we have the sequence really, there is no issues, like in the past we have with hybridomas, with the clone, there's no viability issues.
Yes.
Yeah, and then we can always produce more, and it's fully scalable. So this is really convenient. And so this is about reproducible data. And as I mentioned, we really take care of the characterization and the validation, and that the antibody is working on the right application. This is an example of some of the antibodies that we have for diabetes. This is anti-PDX1 and for other metabolic diseases as well. So we have here a beautiful staining of mouse pancreas performed by immunohistochemistry. So this is for a mutant system, for this is a mouse pancreas. We have also for a human pancreas as well. And this antibody is working on other applications, working on ICC, immunocytochemistry, where we are using, in this case, not primary, but we're using a cell line, which is the beta TC6. It's a mouse pancreas insulinoma beta cell line, which shows positive staining, positive nuclear staining, which is absent in negative control. In this case, this is a NIH3D3 cell line. And in addition, we have other controls, such as no secondary antibody control. And sometimes we also use isotype controls. This antibody in particular is working for other applications like immunoprecipitation and Western blot, also with lysate of this beta TC6 cell line.
Very clean.
It's very clean, sharp staining, and importantly, we know we are adding the right controls, and we don't have any specific staining or any specific bands. So we characterize through different applications. And we are validating with the knockout, as I mentioned, this genetic system to look at the specificity. So this is another example of some of the reagents that we have for diabetes and for metabolic diseases, anti-glucocorticoid receptor antibody. And I pick up this one in particular because, just to illustrate, for some of the knockout validation that we're using, we even use different cell lines with different backgrounds. Just showing one, we have more cell lines in which we knock out the glucocorticoid receptor. In this case, we have a HeLa and we have the A549 cell line. and both different cell lines show nice knockout depletion of the anti-glucocorticoid receptor.
Yeah.
Again, is very clean system, and that give us confidence and show us that our product is really specific. And again, just showing that it works on different applications, and that we use, sometimes we need to use treatments. In this case, we're using dexamethasone to induce the translocation, the nuclear translocation of the glucocorticoid receptor. And this is after 2 hours. We can see that it's now have a nuclear staining by ICC, and it's also working by flow cytometry or by immunohistochemistry. So knockout validation in one is really our gold standard. We are trying to now implement this validation methodology for many other reagents. So we are working on it. We believe is really important in reagents. And but we're only not we're not only supporting diabetes research by antibodies. We also have a wide range of products on kits and assays, and I encourage you to look at the website for these metabolic diseases. We have a nice, beautiful pathway map with the different proteins involved, and where we suggest different kits and reagents as well. So I just pick up a couple of them, like human insulin ELISA, and this is a single-step ELISA, really simple, straightforward. You incubate your sample, which can be either, what samples you have in your hands, like in this case, serum or plasma, and you incubate 1 hour, and then you add the substrate, and in 90 minutes you get your results. Again, we're using the same validated antibodies for these ELISAs and for these kits. And then another example, this glutamate decarboxylase assay. So we measure activity of these enzymes, and we measure with different readouts, so we can get the activity, but also kinetics. And again, this is just an example. We have hundreds of different kits that are suitable for diabetes research. Just mentioned some of kits, ELISAs. I also gave you a couple of examples of reagents that have been knockout validated. We have many more platforms for multiplexing, for instance, Fireplex, and knockout cell lines that you can use for your research. So we have a really wide range of products of interest for diabetes. And just I would like to end up with these slides, and just to mention that we are really committed to help and improve scientific reproducibility, and this goes beyond technological advances. So for example, we organized, and this was last June 2020, sorry, we organized the Reproducible Science Week.
That's fantastic.
That was really great to have so many good feedback, and scientists contributed. And the idea was to champion, the best practice to overcome reproducibility crisis. So it was really like to establish some kind of guidelines. There was something similar to what I show with these pillars, on that, just making sure we have the right controls and things like that. So these kind of initiatives, I think, that are really important also to raise awareness within the scientific community. Something else that we are now being involved on is this Icaros. We are now contributing and helping. This Icaros is an independent open science organization. It's a company actually for antibody characterization. So
and they're based in Canada. I think we have a couple of people from Icaros here. I'd love to have you at the very end, maybe just engage a little bit.
Okay, yeah, that's fantastic. I think it's a great, really, initiative, and that we finally are having, we're having these kind of platforms, and that we can really collaborate and contribute. And what the Abcam is doing is really committed. We are exchanging materials, reagents, and cell lines for validation, and Icaros is really creating these reports where they compare side-by-side antibodies, and that gives really that moves this reproducibility, that gives more confidence in the antibodies. And I think something just to briefly mention, I think this has been appreciated in the community that Abcam is doing such an effort in improving scientific reproducibility. And as a result of that, Abcam has received a couple of awards from Site Up in 2020 and 2021. So we are really proud of that and these initiatives that the company is taking.
Yeah, no, that's fantastic.
Yeah. And with that, I would like to end this presentation, and I will be happy to take any question.
Yeah, I would definitely encourage, audience members to kind of dive in here and, maybe just, I see Carl LaFlamme is on here and Shaitan Reina. If you guys would like to sort of add some value to the conversation, that'd be wonderful.
Sure. Why don't I start, since I'm not a scientist, so my value will be pretty fast. But we've partnered with Abcam, who's been tremendous in providing guidance, reagents, both knockout cells and antibodies to our tests. We're all open science. Everything is up on our website, icarus.com, linked to Zenodo, our Zenodo community, where we've posted, I think, Carl will correct me if I'm wrong, I think we're up to 20 characterization reports for antibodies targeting largely neuro proteins at this point. But we're really looking to expand. The stretch goal is to cover the entire human proteome. And right now we're doing Western, IP, and IF. But we'd love to just keep expanding. All reports public, all data public. We have a monthly meeting with all of the companies participating where we show them our data before it goes up. They, critique our science. We take that feedback. We rerun where appropriate. But really, this is a community effort. So we'd love to just keep engaging with more antibody manufacturers, more scientists, because we're a community resource. So appreciate the invitation today to come and attend.
For sure. Yeah, no, we really celebrate what you're doing. And in fact, we're kind of working with DKNet to try to expand that open science community and also invite people to really get on board the fair data train. So because, NIH here in the US is implementing the mandates in 2023, and that'll be very important for sort of all parties engaged to really understand that reproducibility is kind of like the cornerstone of what we're talking about when we talk about fair data and all those things that are coming down the line. Carl, would you like to say a few words?
Yes, first, Patrice, I don't think we ever met. It's actually very good to meet you, and very great, very good presentation. I was wondering if it's okay if we go back to maybe slides 6 and 7. I might have a few questions.
Absolutely. Let me just share my screen. 6 or 7, you said?
We can start with slide 6.
There we go.
All right.
Okay, so a few questions. So regarding, let's see the 5 pillars for antibody validation that was published in 2016. One of them was, do you get a signal from your antibody that correlates with a good RNA expression in your tissue of interest? So if we look at the IHC figure on the left, IHC analysis of paraffin embedded mouse pancreas tissue. How do we know that the signal that we're observing here with this antibody is actually specific for PDX1?
Yeah, well, so it depends. I mean, for this in particular, we run different controls. So we have different tissues. So we have what we call this Atlas microarray. And so we screen and scan tissues from different organs like liver, heart, lungs. And so we really make sure that the expression pattern is as we expected based on literature, basically. And sometimes we correlate it with some transcriptional expression that is available in some databases. And if possible, we run some controls like knockout or knockdown whenever this is possible. For this particular, I don't think we have this genetic validation, but we did have a validation for screening in different tissues.
IHG is so tough in terms of validation. Something that we might try to implement, and I would love to have Abcam's kind of feedback, would be to use knockout cells as a cell pellet and do IHC on on these cell pellets and try to have whether this antibody, for example, will give a specific signal or not.
Yeah.
That's something we'll try to do as well. But
Yeah,
I understand IHC is very tough.
Yeah, no, you are absolutely right. And this is an excellent point. This is something that we are implementing. So still some sort of pilot assays for some of the high value targets. We have these pellets and with the cell line. So we are kind of testing this protocol at the moment.
Interesting.
Yeah, but for now, we are just relying on this tissue microarray and really literature, transcriptional data when available. Yeah.
Okay, and then if we look now at the IF figure, so we're looking at beta TC6 cells compared to NIH333. So I understand that NIH333 do not express PDX1, that's okay, but there's probably 2,000 different proteins that are present or differently expressed between these two cell lines. So how do we know that the signal that we see in red in TC6 is really specific for PDX1?
Well, sometimes we do. I mean, we do have some stains with some other markers, so we always try to correlate and to find this co-expression of the markers. So yeah, we have we need to test the specificity by testing different cell lines, and we really based our data, positive or negative, based on publications, and in the same case as in immunohistochemistry, we use the models when they are available by using stimulations to see if we can get upregulation or induction, or when possible we can use these genetic systems for knocking down or knocking out.
Okay. Exactly. I always feel it's tough because this is something at some point that I wanted to do as well. So we had this knockout clone by Western blot. We could tell that this knockout, was indeed a knockout. But then in IF, we couldn't identify any antibodies. If though we screened, I don't know, 20 antibodies by immunofluorescence, they all failed. So we screen a bunch of cell lines and find that cell line expresses very high amount of that target. And the question was, okay, let's use this cell line with very high expression compared to a knockout cell line, exactly like you're doing here, and let's do an IF. It worked in the sense that there was a specific signal in that wild type line. However, the fact that all these proteins are differently expressed between two different cell types makes it very hard, I think, to make any conclusion. But I totally understand. The strategy.
Yeah.
And maybe
sometimes is sometimes one. These are our challenges, right? To find the absolute final and decisive assay to. So we are doing as best as we can by using all the I don't know, by using different cell lines, for instance. Now we are displaying only these two, like positive and negative, but when we review this data, we normally get at least 2 or 3 positive cell lines or negative to really make sure that it is expressed on the cell that is supposed to be expressed, and with the pattern is supposed to be. Is it nuclear or is it in the cell membrane? And as I said, it is inducible. Yeah. But I mean, we are happy always to hear any feedback and suggestion on,
Maybe
ideally.
Tell me. Yeah, go ahead.
No, I mean, for tricky targets, what we try to do, and if we have any doubt whether it's specific, whether the signal we are getting is really from from the protein that we expect. We really reach out to collaborators that are working sometimes with a knockout mouse model. And this is actually the case for some of the antibodies that we are developing now for diabetes.
Awesome.
And, you know, we just send them the clone, they tested with their knockout mouse material, and we review the data together, and then we sometimes incorporate the data in the dataset.
Awesome. Awesome.
Yeah, and you
Very
can
good.
see that actually is sort of another nod to the power of the community that you're engaged with and embedded with. Is that, you know, if you come across some kind of you know, issue, you can sort of reach out to these constituents and they can validate what's happening, and it's very collaborative.
Yeah, absolutely. No, this is probably one of the most important pillars, together with these genetic models for validation. This is the collaborative network.
Yeah.
And this is absolutely critical for us to develop good antibodies. Yeah.
Indeed. Indeed. And maybe one last question regarding the next slide.
Right.
So by IF, you use the treatment with dexamethasone, and that's fair, to see the expression of here it's glucocorticoid receptor. That's okay. But then in Western blot, you do not use any treatment. So how come by Western blot you don't need a treatment, but in IF you do need it?
Well, we do have some expression on a naive in the naive cell line. You know, we do have some expression. You can see that is the staining on the cytoplasma. Maybe it's not very obvious, but we do have expression. I mean, it is expressed on the cell. It's just this translocation that we are looking at after the treatment, and that we can see that is moving from this cytoplasm location into the nucleus, and that we have a high an upregulation of the protein expression. This is what we're looking at when we do the treatment by IF. And so on the wild type, we are able to detect it, but probably if we do a treatment on the wild type, we should probably see maybe higher expression.
So since you have an HeLa knockout, why not using it by IF? So use your HeLa knockout, treat it with dexamethasone, and show that the IF signal is gone.
Yeah, absolutely. That's a great idea. Sometimes we try to do the same strategy for all the applications. And even for I'm showing now a flow cytometry data with the HeLa cells that are not treated. And because sometimes, you know, the way we do antibodies sometimes is we validate antibodies for 2 applications, and then we validate them afterwards. So that means that then we run, for example, flow cytometry, or in this case, we run ICC or IF. So sometimes they don't have exactly the same treatment, or sometimes it's just not necessary. What we try to do is, as much as we can, is just keep it consistent. If we do a treatment, it's nice to see it in flow cytometry as well, for instance. So we try to do that whenever it's possible, indeed. Yeah.
I see. Thank you very much. Thank you.
You're welcome.
I have to just add, I think that this kind of conversation is, you know, really important as you go back and forth on the finer details, the granular pieces, because I think it's so valuable that I just think the audience can see that the level of care that both groups, Icarus and Abcam, pay, the level of attention and care that is paid to developing these best-in-class antibodies, but also in, you know, again, reaching out to the community to add value to making this happen. So I think it's what you have here is a powerful, you know, symbiosis, I think. And I'm really looking forward to seeing how these 2 entities drive, you know, better antibodies into the future. I think you guys are doing something really significant and really important, and appreciate you all joining today. And Patricia, thank you again for your excellent presentation. It was really great.
Thank you so much for the invitation. It was my pleasure to
Yeah.
with you guys.
Fantastic. I look forward to meeting you all again in the future.
All right. Thank you.
Thank you.